therefore PCR is one of the tools most responsible for revealing to scientists the enormous diversity of the microbial world, and making it clear that microorganisms that have been cultured make up but a tiny fraction of the microorganisms in the environment. Because of this ability to amplify and analyze DNA without having to grow the microorganism, PCR has also taken over a very important role in diagnostic microbiology. It has also been used in DNA fingerprinting, a powerful forensic tool that permits identification of individuals or relationships between individuals from very small samples of their DNA. DNA fingerprinting in Chapter 31. PCR can also be coupled with reverse transcription and a process called RT-PCR. This can be very useful when one wants to make large amounts of DNA using RNA as a template. In vitro, site-directed mutagenesis. Methods of in vitro DNA manipulation have opened up a whole new field of mutagenesis, whereas conventional mutagens act as a random by use of synthetic DNA and recombinant DNA techniques. It is possible to introduce mutations at precisely determined sites on genes. This process is called site-directed mutagenesis. Proteins made from strains carrying such mutagens can be expected to have properties different from those of the wild-type proteins, properties that may in some cases be predicted from knowledge of protein structure. Site-directed mutagenesis. The first approaches to site-directed mutagenesis included treating transducing bacteriophage with chemical mutagens and then infecting bacteria screening for mutants. Such a strategy had the effect of increasing the mutants rate in a limited region of a genome. New techniques are now available that allow the geneticist much greater specificity. A specific base pair in a gene can be changed to another base pair. Some of the techniques are simple and very powerful. The basic procedure is to synthesize a short oligodeoxyribonucleotide containing the desired base change, and to allow this to pair with a single strand of DNA containing the gene of interest. Pairing is complete except for a short region of mismatch. Then the short single-stranded fragment of the synthetic oligonucleotide is extended using DNA polymerase, thus copying the rest of the gene. The double-stranded molecule obtained is inserted into a cloning host by transformation. The mutant selected by the procedure already described. The mutant obtained is then used in production of the modified mutant protein. One procedure for site-directed mutagenesis is illustrated. A vector. It can be convenient to begin with the gene of interest cloned into a single-stranded DNA vector. A widely used vector for site-directed mutagenesis is bacteriophage, M13. We shall discuss M13 as a vector in Chapter 15. Cassette mutagenesis and gene disruption. Because of the large number of restriction enzymes commercially available and therefore the large number of different DNA sequences that can be cut, it is easily possible to find several different restriction sites in the gene of interest. If the sites for the appropriate enzymes are not found in the gene, or at the precise location required, they can also be added by site-directed mutagenesis. If restriction sites are close together, the intervening DNA fragments can be cut out and replaced by a synthetic DNA fragment in which one or more of the bases have been changed. These synthetic fragments are called cassettes, or cartridges, and the process is known as cassette mutagenesis. Insertion mutations can also be generated by simply inserting a cassette at a single site. When using cassettes to replace sections of genes, the cassettes are typically the same size as wild-type DNA fragments. However, the cassette used for making insertion mutations can be almost any size and can even be an entire gene. In fact, cassettes encode proteins that confer particular antibiotic resistance on the host are commonly used. The type of cassette mutagenesis is used in a process called gene disruption. This process of gene disruption is illustrated. In this case, a fragment carrying a gene conferring kanamycin resistance to cassette is inserted at a restricted site in a cloned gene. The vector carrying this mutant gene is then linearized by being cut with a different restriction enzyme, and the linear DNA is transformed into the host with kanamycin resistance selected. The linearized plasmid cannot replicate, and so resistant cells likely arise by homologous recombination between the mutated gene on the plasmid and the wild-type gene on the chromosome. The cells have not only gained kanamycin resistance, but have also lost the function of the gene in which the kane cassette was inserted. Therefore, these mutations are also called knockout mutations. This process is similar to searching for insertion mutations made by transposons, but in this case, the geneticists choose exactly which gene will receive the mutation. Note that gene disruption in haploid organisms yields viable cells only if the disrupted gene is unessential. Gene disruption experiments are now used as one mechanism to find out whether a gene is essential. Methods of obtaining gene disruption have been developed for higher organisms, including mice. Here we go. The bacteria chromosome. The three mechanisms of genetic exchange described in chapter. The three mechanisms of genetic exchange described in this chapter, transformation, transduction, conjugation, can be used to map the locations of various genes, actually mutations in genes, on the chromosome. In Escherichia coli were mapped to a particular region of the chromosome using conjugation. By using high-frequency strains with origins at different sites, it is possible to map the whole bacteria gene complement. Conjugation does not permit ordering genes that are closely linked. Therefore, generalized transduction was used for more fine structure mapping of E. coli chromosome. Bacteriophage P1 can carry fragments of DNA equivalent to about 2 minutes on the map and has proved very useful for mapping genes. The genetic techniques that were used were dictated by the efficiency with genetic transfer occurs in the organism to be studied. Transformation is a very inefficient process in E. coli, but is more efficient in other organisms and has proved an effective tool for mapping genes in bacillus. Although all these classic techniques are still useful for the characterization of construction of strains, they have largely been supplemented in constructing genetic maps for molecular cloning. Section 10.14 Restriction Mapping, Section 10.12 and DNA Sequencing, Section 10.13 which have revolutionized the study of the genome organization of prokaryotes and eukaryotes. And this is the end of the chapter. It's been 3.5 hours now, and I am exhausted. So, thank you and take care.